Hello, everyone. My name is Asmann. I'm an associate professor of biomedical informatics in the Department of Quantitative Health Sciences at Mayo Clinic. Today, I will talk about the bioinformatics of neoantigen discovery and vaccine design. First, what are neoantigens? As shown here in this perhaps overly simplified diagram, proteins are degraded and the peptides are transported into the ER. In the ER, peptides are loaded onto MHC molecules, and the peptide MHC complex is then transported to the cell surface and presented to the T cells. T cells read each peptide to determine if it is foreign and if it's necessary to mount an immune response. As we know, there are many proteins expressed at much higher levels in tumor compared to normal tissues. The so-called tumor-associated antigens, or TAAs, are abundant. In addition, tumors' somatic mutations will generate mutated peptides. These are the new antigens or neoantigens. Both TAAs and the neoantigens can potentially solicit an immune response. Many years ago, Dr. Keith Knudsen, a immunologist at Mayo Clinic, discovered that patients with breast cancer demonstrated elevated T cells and antibody immunity to several tumor-associated antigens. These three figures show that elevated TFH, CXCL3, and the TH1 are associated with better survival. Therefore, the immune system actually naturally responds to tumor. However, to be clinically apparent, the tumors must have evaded the immune surveillance. This is because, as shown in the figure to the right, many tumors express cell surface immune checkpoint proteins and disguise as normal cells. Immune checkpoint blockade, such as PD-L1 or PD-1 inhibitors, works by jamming that normal signals so the immune system can see the tumors again. However, the efficacy of ICB remains limited and is well below 20% today. Therefore, even though immune checkpoint blockade therapy has revolutionized cancer treatment, novel immunotherapy agents are being actively developed to overcome resistance and to improve efficacy. This is another study by Dr. Knudsen's lab showing that tumor-associated antigens enhance the efficacy of immune checkpoint blockade. As shown in figure C, the combination of anti-PD-1 and the C3L3N1 TAA vaccines inhibited tumor growth for significantly longer period and resulted in much better survival, as shown in figure D. I already introduced the concept of cancer neoantigens. Just to repeat, neoantigens are mutated peptides presented by MHC molecules and recognized by the immune system. Because somatic mutations are more cancer-specific, neoantigens are consequently highly cancer-specific and potentially more immunogenic compared to TAAs. Here's a brief overview how neoantigen works. The tumor cells, like the one in the center of this diagram, present neoantigens by MHC to T cells. In addition, antigen-presenting cells such as the dendritic cells, the blue large cells look like starfish, can also take up and present neoantigens. Together, this process leads to adaptive immune responses that kill cancer. This is how endogenous neoantigens work. However, the endogenous neoantigen levels are often very low. So if we could identify what neoantigens a patient's tumor has and supply the extra dose by vaccines, these synthesized neoantigens will train the immune systems to look for tumor. And when ICB removes the tumor surface immune checkpoint signals, the trained immune cells can readily target the tumor. So at Mayo Clinic, we decided to build a neoantigen vaccine therapy program. As I mentioned, neoantigens come from somatic mutations, including single nucleotide changes and small insertions and deletions in genomic DNA. Also, aberrant splicing isoforms of the RNA transcripts and genomic rearrangements, such as translocations, inversions, and copy number alterations that lead to gene fusion. It is worth mentioning that not all neoantigens are the same. Some only harbor a single amino acid change. Others, due to protein fusion or translational frame shifting, can be very different or entirely new compared to wild-type proteins, which are generally more immunogenic. Later, I will describe some of the algorithms we developed in-house to identify these potent neoantigens. The features that constitute of high-quality neoantigens from left to right, top to bottom include, first, high degree of non-selfness. For example, neoantigens from frame shift indels have higher degrees of non-selfness compared to peptide with a single amino acid change. Second, tumors are known to have immune selections during proliferation and progression, and lose clonal neoantigens in new subclones. So if a neoantigen is from passenger mutations of a gene critical for tumor survival, it is more resistant to neoantigen loss. Number three, neoantigens that can be presented by multiple HLA study. Genes harboring neoantigen modulations is in stable genomic regions with lower likelihood of copy number loss, will produce stable neoantigens. And number five, studies also found that a minimal cell fraction is required to trigger immune response. So neoantigens from a small subclone may not be as effective. And the last, neoantigens need to be expressed at moderate or high levels. Neoantigens are highly personalized or patient private. For example, when we studied about 850 breast cancer patients, 99.75% of the neoantigens were only observed in either a single patient or less than 1% of the patients. And towards the top, only 18 neoantigens were shared by more than 5% of the patients. Our findings are consistent with previous reports in melanoma and other tumor types. Because of this, neoantigen-directed immunotherapy needs to be highly personalized as well. Neoantigens can be used to treat cancer in the forms of vaccines, cellular therapies, in combination with ICB. In addition, neoantigens are also biomarkers that predict ICB outcome and patient survival. With the lack of accurate measurement of neoantigen load, however, tumor mutational burden has been used as a surrogate. We have published that neoantigen load actually is a better biomarker than TMB. This 2015 science paper grouped the tumor's ability to produce neoantigens based on TMB, as shown in the y-axis. The cancers in green box with high TMB, such as melanoma and lung cancers, are predicted to frequently form neoantigens, while the tumors with low TMBs to the left such as some of the hema malignancies only occasionally form neoantigens. Here, I highlighted breast cancer pointed to by the red arrow, which has medium levels of TMB, to show in the next few slides that TMB underrepresents neoantigen load and is not an accurate surrogate. TMB is calculated as number of protein-altering mutations per megabase of coding DNA, which comprises of SNV, indel, and splice site mutations. First, the ratios of mutation versus neoantigens are often 1 to many. Second, both genomic rearrangements and RNA de novo splicings are not counted towards TMB. An earlier Nature Genetics paper showed that cancers in ovary or breast are C-type, mostly driven by genomic rearrangements, but not small mutations. Our own work to the right showed that not all splicing events had associated DNA mutations. For 16 lung cancer patients with known MET gene exon 14 skipping events, 9 came from de novo RNA splicings. Only 6 resulted from DNA splice site mutations. Therefore, TMB underrepresents neoantigen load. This Nature Genetics paper from 2019 showed that higher tumor mutational burden predicts better overall survival after ICB therapy in multiple cancers, highlighted in red box, but not in breast cancer patients. I highlighted the breast cancer p-values here by the red arrow. However, we published that lower neoantigen load, not TMB, is a biomarker for worse overall survival in breast cancer patients, independent of other clinical predictors, such as tumor infiltration, tumor infiltrating lymphocytes, breast cancer subtypes, tumor stage and age at diagnosis. Again, supporting the notion that neoantigen load is a more accurate biomarker. As shown towards the bottom of this slide, neoantigen peptides can be directly identified by mass spectrometry analysis of MHC ligands. However, this process can be laborious and requires large amount of tissue. Today, neoantigen detection is mostly done by bioinformatics analysis of tumor DNA and RNA sequencing data in 3 steps. First, patient-specific MHC is genotyped. Second, potential neopeptides from mutated proteins that are expressed in tumor cells are generated. And then bindings between MHC and neoepitopes are predicted. We developed an in-house bioinformatics framework called RealNeo. Here is a diagram of the version 3 of the pipeline, which is a combination of public tools and our own novel algorithms. This in-house pipeline enables us to quickly incorporate new knowledge and new functionalities and perform QC and the prioritization of the neoantigens without dealing with the black box problem. So for clinical trials, FDA is okay with us using research-grade data and analytics for compassionate use cases and early phase clinical trials. But if we want to conduct neoantigen-related trials in a bigger scale, having a pipeline with black boxes won't be okay. So first, the sequencing reads are used for somatic mutation discovery and the tumor MHC or HLA genotyping. Second, we predict mutated peptides resulting from the mutations. And the third, neoantigen predictions, annotations, and prioritizations were performed. I will describe several components of RealNeo in more details, including the algorithms to identify splicing neoantigens, and one of the new features in RealNeo version 3, the identification of disruptions in the antigen presentation pathway. So first, the identification of aberrant splicing neoantigens. First of all, there is a category of DNA mutations located in splice sites that can lead to aberrant splicings. As shown in the figure to the left, the quantity of these mutations are on the par with indels and gene fusions. Using TCGA breast, liver, and the 2 lung cancer types as examples. However, these splice site mutations are not the only cause of aberrant splicing. As shown in the figure to the right, which I already showed this figure in a previous slide. In the example of MET exon 14 skipping, de novo RNA splicing is responsible for the majority, 9 out of 16 of the aberrant splicing events. RealNeo is capable of detecting both categories of highly immunogenic aberrant splicing neoantigens. Here is the algorithm for identifying splicing neoantigens from tumors with DNA mutations in splice sites. As shown in figure A, we start from DNA mutations and predict splicing donor or acceptor site gains and losses use a published tool called Splice AI. After survey of close to 12,000 TCGA cancer cases, 13 distinct profiles were detected as shown in figure B, some of which are very frequent and others are more rare. The top 7 profiles represent 95% of the splicing events, and the simplest profile pointed to by the red arrow has a single loss of the original splicing acceptor site. This simple profile, however, as shown in figure C, generated rather complicated 3 different splicing alterations. Depending on the location of the acceptor loss, either a middle exon or in the last exon of the gene, it could lead to intron retention or exon skipping. RealNeo computes all potential splicing alterations from all 7 profiles. Most are way more complicated than this simple example. So once we obtain the putative splicing isoforms, we confirm that these splicing products are expressed by quantifying the expression as shown by the y-axis in figure D, and by calculating a z-score as shown by the x-axis of the figure D, using the entire TCGA cohort as the reference, assuming that individual specific aberrant splicing events are rare. Here is a summary of aberrant splicing events and the neoantigens from DNA splice site mutations, site changes. For example, the combination of acceptor loss and donor loss in pink towards the bottom that gave rise to predicted splicing events, the confirmed prediction by both expression and z-score, and the number of neoantigen candidates in TCGA breast, liver, lung adeno, and the squamous cell carcinoma. To the right are the counts of different types of splicing events, such as in-frame or frame shift exon skipping. I'll let you stare at the slides for a couple more seconds. Next, here's how we detect de novo RNA aberrant splicings. As shown in the figure to the left, the amount of de novo RNA splicings are comparable to the level of splice site mutations. To the right, from the top, we start with the junction support reads. For example, in a sample, we might detect both exon 1 and 4 and exon 1 and 5 junctions. Next, we use a z-score threshold assuming these events are rare, and in addition, require that the novel transcripts are expressed and the fraction of the novel junctions are at least 10%. Next, we eliminate E1, E5 if there are evidence showing the so-called E1, E5 junction reads actually also support E1, E4. Then we do another round of transcript quantification to make sure that the novel splicing events are indeed expressed. The novel transcripts will then be translated into k-mers from MHC binding prediction to identify neoantigen candidates. As shown in the figure to the right, the numbers here confirm that about 40 to 50% of the aberrant splicing events are detected by both approaches towards the top. The majority of the splicing events are de novo, without splice site DNA mutations, towards the bottom. We suspect that there are a lot of false positives in this group, and we are further optimizing our algorithm using long-read RNA-seq data. There are also a small percentage, around 5% in the middle, of the splicing events that are only detected by the splice site mutation approach. RealNeo is fairly accurate in predicting neoantigen. Here I show laboratory validation of results of novel neoantigens from aberrant RNA splicings. Please ignore the fusion neoantigen results for now. For validation rate, the validation rate was almost 100% for neoantigens that bind to HLA-0201. Some even bound stronger than the positive control, which is a peptide from flu virus. In version 3 of the RealNeo, we added several new functionalities, including evaluating the antigen presentation. As shown in the figure to the left, we identified somatic and germline mutations that potentially disrupt the antigen presentation pathway genes and calculated an impact score. As shown to the right, this is the distribution of the impact scores in TCGA cohort. It's a bimodal distribution, indicating that the patients can be grouped into two categories: those with higher impact scores with disruptions in the antigen presentation pathway, and those with lower impact scores with less disruption. And one of our patients, PT453, who achieved partial response to the neoantigen vaccine therapy, belong to the not significantly impacted category. This is mutations alone in the antigen presentation pathway. We're actually in the process of combining mutations and expression levels to compute a composite score. At Mayo Clinic, we are conducting both single patient and phase 1 trials. One of the single patient IND example is a 26-year-old renal cell carcinoma patient. 21 neoantigens were identified from the primary tumor as well as the liver met, and the patient achieved partial response 4 months after the vaccine ICB combination. We have a phase 2 trial in development to test nanoparticle delivery of the polypeptide neoantigen vaccines. We also developed different neoantigen-derived cellular therapy products, including TCR-T and the biospecific antibodies. I'll use the results from the single patient IND renal cell carcinoma neoantigen trial to demonstrate the accuracy of RealNeo. 14 out of the patient's 21 neoantigens are HLA-0201 binders, arising from SMV, indel, fusion, and splicing mutations. We were able to validate in the lab 13 out of the 14 nominated neoantigens, which is a 93% validation rate. In addition, the real Neo-nominated neoantigens had significantly higher processing and presentation probability scores compared to the random peptides, as shown in the left. We obtained post-vaccination PBMC from the patient and detected T-cell activities after peptide simulation using ELISpot assays. The 21 neoantigens were manufactured and pooled into 5 vials. All 5 pools resulted in T-cell activities compared to control, and 3 pools with much higher T-cell activities. Here is the clinical course of the same patient. Neoantigen vaccines were delivered starting at day 650 in combination with nivolumab and an anti-PD-1 drug. CT scans at day 710, 800, and 920 showed continuous shrinking liver metastasized tumors. The patient achieved partial response on day 800, 4 to 5 months after the vaccination started, and the tumor continued to shrink after. On day 920, we performed a biopsy showing mostly dying tumor cells with substantial tumor infiltrating lymphocytes, as shown in the figure to the right. Currently, we are actively working on the following areas. 1, to incorporate immunogenicity and then natural processing scores into real neo. We're working on additional source of neoantigens, including non-coding region expression, microexons, and RNA editing. We're developing algorithms for B-cell epitopes dissection. We're working on mRNA vaccines design because so far we only use peptide vaccines. And finally, we're also applying machine learning and AI models for patient selection and outcome prediction. Last but not the least, I'd like to thank my colleagues and the funding we received from Mayo Clinic, and thank you.